Good morning once again, everybody. Welcome to today's webinar, Coral Reefs in Crisis, a mass bleaching event in the Caribbean. Our speakers today are Dr. Craig Dahlgren and Dr. Valeria Pizarro. Dr. Craig Dahlgren is our Executive Director at the Perry Institute for Marine Science, an internationally renowned tropical marine expert with more than 30 years of experience working in the Bahamas and the Caribbean. Craig has a deep understanding of topics ranging from mangrove and coral reef ecosystems to the efficacy of marine protected areas and the population dynamics of traditional and emerging fishery species. His research has been published extensively in peer-reviewed journals and has had immense influence on marine resource management and policy in the Bahamas, the Mesoamerican reef system, and beyond. Dr. Dahlgren's current work focuses on commercial fishery species like the Nassau grouper, queen conch, and spiny lobster, as well as emerging fisheries like sea cucumbers, parrotfish, sponges, and gorgonians. His pioneering research also spans the fields of coral disease and treatment, and mangrove restoration, which we are going to learn about more today. Dr. Valeria Pizarro is a biologist who studies the biology and ecology of corals and coral reefs. She started studying coral reefs in her natal Colombia at the island in San Andres while studying her master's, where she focused on brain coral population dynamics. After that, Dr. Pizarro worked on the design and implementation of the Sea Flower Marine Protected Area System in the Western Caribbean, and started her doctorate studies at Newcastle University in the UK on Caribbean coral connectivity. Her research interests include coral reproduction, population dynamics, connectivity, and coral restoration. In 2019, Dr. Pizarro worked in the Bahamas Coral Innovation Hub at Eleuthera, a project aiming to scale and improve the restoration of Bahamian coral reefs. Dr. Pizarro was a professor at the Jorge Tadeo University in Colombia for 7 years, teaching undergraduates and graduate students. She also has extensive experience on other coastal marine ecosystems, such as mangroves and seagrasses. Her other interests are marine policy, community engagement, and biodiversity offsets. During the last 6 years, Dr. Pizarro has been an advocate of a Colombia reef named Varadero, which could be dredged for a new shipping channel at Cartagena. Her campaign to save this reef has included its nomination as a Hope Spot for Mission Blue. Before we start, we do have a few housekeeping rules. Please note that this webinar is being recorded. Also, a reminder to keep your microphone muted during the session. Also, afterwards, there will be a Q&A session where you will have the opportunity to ask your questions. You can also add any questions that you have into the chat, and after our speakers are done, an audience feedback link will be shared in the chat for your feedback. So, without further ado, I will now welcome our speakers to begin their presentation. Thank you.
Thank you, Danielle. I'll get started here. We're going to be talking today about the current bleaching event that's happening for coral reefs throughout the Caribbean, and we're going to go into a deep dive into the bleaching event and its implication in the Bahamas, where we're currently doing quite a bit of work on it. Before we get started in that, though, just a little bit about Perry Institute for Marine Science. We're a nonprofit organization that for over 50 years has been working on research and conservation of tropical marine ecosystems in the Bahamas and throughout the Caribbean, trying to make an impact on the sustainable management of marine resources and habitats in the region. Our main focal areas include coral reefs, fisheries, coastal habitats, and the people that these reefs, fisheries, and habitats affect. Our roots are in the Bahamas. We've been based in the Bahamas for over 50 years, and for a good chunk of that time, operating a marine lab on Lee Stocking Island. In the Exuma Keys, the Caribbean Marine Research Center, from about 1984 until about 10 years ago. And now we're working throughout the islands of the Bahamas and have projects in now 11 different countries in the Caribbean. So we definitely are getting involved on a more regional basis. And for today's webinar, I'll turn it over to Dr. Pizarro to start with, to go over a lot of the biology and ecology of coral reefs, what coral bleaching is, what some of the other threats that corals are facing, and then looking at the current marine heat wave that's affecting reefs throughout the region. Then I'll take over to talk a little bit more about the Bahamian experience historically up to the present with this bleaching event, what some of the implications of that are, and what we can do about it, where we do have some hope. So I'll turn it over to Dr. Pizarro to get started.
Thanks, Greg, and thanks, Danielle, for the introduction. Good morning, everyone. Thank you for being here. So yeah, I'm going to start then with just some very important but general information about coral reefs and corals. So coral reefs are coastal marine ecosystems and are some of the most diverse ecosystems in the world. Their importance rely on what these ecosystems provide to millions of organisms, but more importantly to us humans. These are known as ecosystem services. Coral reefs protect coastlines from storms and erosion, provide jobs for local communities, offer opportunities for recreation. They are a source of food for billions of people, and they are a source of new medicines. Over 500 million people depend actually of the reefs, not only for food, but as well income and protection. Fishing, diving, and snorkeling on and near reefs add hundreds of millions of dollars to local businesses, and the net economic value of the world's coral reefs is estimated to be nearly tens of billions of US dollars per year. And coral reefs are known as the tropical rainforest of the sea. Specifically in the Caribbean, approximately 31 million people live along the coastline. It holds 10% of the world's coral reefs and are home to more than 12,000 species. These ecosystems are built mainly by stony corals, so they are like the trees of the seas. But what are stony corals? So they are in fact animals. While having the appearance of vibrant rocky outcrops, these animals most closely relate to jellyfish and anemones. Almost all corals are colonials, and what this means is that they are composed of hundreds of thousands of individual animals called polyps. They secrete a rock chalk-like substratum that is actually calcium carbonate, and it's an exoskeleton that they grow around themselves for protection, and they continue to secrete these calcium carbonate throughout their life, and that way they grow as a colony, but they are starting like building and continue to build coral reefs. Each one of these individuals that are called polyps, they have a stomach that opens at only one end, and this opening, called mouth, it's surrounded by tentacles. These tentacles are used by the polyps for defense, to clear away any debris, sedimentation that just go on top of the coral, but they also capture small animals for food that is called zooplankton. And although feeding from these small animals, zooplankton, is one of the ways corals obtain food, the main source of food and energy for the corals come from a symbiotic relationship that they have with a one-cell algae called zooxanthellae. This symbiosis is very important for the coral. All these algae, and they live in the thousands inside the corals, they provide all the energy necessary for the coral to live and survive. Even though they can feed on this zooplankton, the zooplankton is not enough food for the coral to live, so they need this algae. This symbiosis is so important that the coral, that they cannot survive then without the algae, and they're known as obligate symbionts. Additionally, every time that you go diving or you go to the water snorkeling and you see a coral, if you see like these brown, green, yellow corals, all these corals, all the colors of these corals are from the algae. The algae gives the color to the coral. And as most of you might know, global changes are affecting our climate and our marine environment. Coral reefs are being affected by many threats that include severe storms, hurricanes, sea level rise, ocean acidification, increased pollution, and of course, increased water temperatures. The potential consequences of higher seawater temperatures are coral disease outbreaks and bleaching events. So now I'm going to explain what is the difference between those two. So coral diseases are often visible as a change in the tissue coral color, but more importantly, on the mortality of the coral. And this mortality can be partial or total. Partial mortality is just part of the coral. And remember, these are polyps that form. So some of these polyps, as the photo that is on the left side of the screen, this white part, they are partial mortality. So those white polyps, they're already dead. But we can have in coral diseases total mortality, meaning that the whole colony will die. The origin of coral diseases, which can be viral or bacteria, is very difficult to determine and is not clearly understood by scientists. Viruses, bacteria, and other microorganisms, or even changes in environmental conditions can contribute to coral diseases. A certain amount of infectious microbes that cause coral diseases are naturally occurring and always present in an ecosystem. However, an event or combination of these events that disrupt the natural levels can encourage coral disease. It is important to understand that even though the most important symbiosis for the coral is the zooxanthellae, actually corals are in symbiotic relationship with a variety of bacteria and viruses and other microorganisms, which are mainly responsible for the coral's high contribution to primary productivity of coral reefs, and any modification on the structure and relative density of the symbiotic microbial communities. All these small organisms that live within the coral might have a critical role on coral adaptation to rapid environmental changes, and as well coral diseases outbreaks. On the other hand, coral bleaching is a stress reaction of the coral animal that happens when they expel the symbiotic algae, the zooxanthellae. So remember that I told you that the corals have, the color of the corals are these, comes from the algae. So anytime that they're stressed, and this stress is mainly related with temperature, but it can happen as well with high sedimentation as well. So with higher temperature or very low temperature, corals start to expel the zooxanthellae and they become bleached. So the tissue of the coral is translucent. So when we have coral bleaching, when we have the white corals that we see when we have a bleaching event, like the photo in the middle on your left side, that means that the coral are alive. They're not dead, they're still alive, but they are very stressed and they are less likely to reproduce and are more susceptible to disease, predation, and mortality. If these stressful conditions that resulted on bleaching continue, the coral will die of starvation, because remember, the algae gives most of the energy and food that they need to live. But if these stressful conditions subside soon enough, corals can start getting the algae back and they can survive and keep growing into coral reefs. So it's very important just to have clear, when we have a coral disease, coral can die and they cannot recover from that, but with coral bleaching, they can recover from that. Okay, sorry. So until the end of 2019, Bahamian coral reefs were relatively in good condition compared to most Caribbean coral reefs. As you can see in the figure, many reefs were in fair conditions by 2019. This data was like taken before, like until around mid 2019, and especially the best conditions were found in all those islands that are located in the south of the Bahamas and southeast. Sadly, at the end of 2019, a deadly disease started to affect the Bahamas. This disease is known as stony coral tissue disease, or SCLD for short. It affects more than half of reef-building coral species, and it spreads very quickly, killing hundreds of corals in a matter of weeks. From our observations, stony coral tissue disease has spread to many of the family islands, to about 60% of Bahamian coral reefs, as you can see in the figure. All the red lines indicate where the disease has been confirmed. And these images just show you how fast the disease kills corals. This is a shallow reef in Lucayan National Park in Bahama. The first one on the top was taken in March in 2020, and the second one in March 2021. So the one that we took in March 2020, you can see some corals that already have the disease, and it started to spread. And in the ones in 2021, just show you how many are remaining left. This is not very easy to see, but in these other two, you can see all the outline corals. This was a shallow reef dominated by brain corals, and all the outline corals are the ones that are alive. As you can see from the one on the top on your right hand, on the right in your screen, and to the bottom, the mortality was really high. We lost actually more than 90% of the brain corals on that area. And here is a figure to show you how some of the most important building coral species that we have in the Bahamas have been affected by this disease. So we have here brain corals, star corals, and the starlet massive coral. The black bars show you the number of colonies per reef site that we were assessing before the stony coral tissue disease was present, and the white bars after 1 year of the disease affecting those reefs. For these species, in average, we lost 75% of the coral colonies. That means that when we were counting 148, we now are counting 35 colonies. And this is like affecting all the community of the coral reefs around the Bahamas where the disease is present. But that information, we wanted to estimate how many coral colonies have died from SCLD. So we know from our AGRA surveys, that is a survey that we do every couple of years around the islands in the Bahamas, just to understand and know the conditions of reefs in the Bahamas, that we have in average 4 colonies per square meter. And we know from some data from the Nature Conservancy that the coral reef area in the Bahamas, it's about 2,162 square miles, or 5,600 square kilometers. So when we do like the calculation, we know that before SCLD was around, we had about 22,000 million coral colonies in the Bahamas, in around all the family islands and reef areas in the Bahamas, with 60% being affected by SCLD by June 2023, before we had the bleaching event that Craig is going to describe you a little more. We know that we had left about 12,000 healthy coral colonies. So we had lost in between 2019 and 2023, approximately 10,000 million coral colonies due to SCLD. And this number is high and it's very shocking. But the truth is that when I was in a talk last week from Dr. Stephanie Shopmeyer from NOAA, and she was presenting the results of the study that they did in a small part of Florida reef tract in the north. And from their calculations, they have lost already from 9 highly susceptible species for SCLD and important in reef building, more than 44 million corals. Now they're doing all the estimations for the Florida Reef Tract, and she said that they are expecting to be actually the number in the order of the trillion with a T corals lost by the disease. On top of this, these years we're experiencing extremely high temperatures, not only in the sea, but in land as well. And this is affecting many organisms and ecosystems, including coral reefs. These high sea temperatures are known as marine heat waves, which are defined as any time oceans are warmer than 90% of the previous observation for a given time of the year. So this year we had really high temperatures that are more higher, as you can see in the figure, the black continuous line, the thick one, that is the temperature that we had this year. So marine heat waves they occur actually almost every year, and they can last for weeks, months, or years, but scientists don't yet fully understand the factors that have caused ocean temperatures to jump this year to these new highs. Certainly, the emerging El Niño event is an important driver. We know from past experience that El Niño event increases global average temperature by about 0.1 to 0.2 Celsius. That is very low. However, this effect tends to come later in the year when El Niño reaches its peak around October and November. Furthermore, we are currently observing two huge heat waves in the North Pacific and North Atlantic, which got underway at the beginning of the year and has intensified and expanded in recent months. This combination of El Niño and extratropical heat waves is driving up global ocean temperatures tremendously. So Caribbean and Atlantic seawater temperatures started to increase in June. And by July, the water temperatures throughout the Gulf of Mexico and the Caribbean were approximately 1.8 and 5.4 degrees Fahrenheit warmer than normal. That is 1 to 3 Celsius. Temperatures in the southern Florida actually are the warmest on record since 1981, sorry. So coral reef scientists and managers use products like this map that you are seeing right now. This map is created by NOAA. And in here, what it shows are the areas where bleaching can occur that are in yellow and orange, that is called like watch and warming, and where it is actually occurring, light and dark green, that are alert level 1 and alert level 2. The Bahamas in general has been in alert level 1 and 2 for over 2 months, which increases the chances of massive coral mortality. And I'm gonna let Craig explain all the rest. Thank you.
All right, thanks, Valeria. So I'll show some similar slides about ocean temperatures, but before that, just want to kind of get in the water and show what some of the bleaching looks like that we're seeing right now. And I'll just show a couple side-by-side comparisons. The first is from a reef off of New Providence Island off Nassau called Bond Reef, where we have photomosaic images of large areas of the reef in January this past year, and then again just about a month ago, in August. On the left side, the January photograph shows what the reef looked like then, dominated by staghorn coral, all of which was fairly healthy. If we look at the August image on the right, though, you can see all of that stands out in white. That's what the bleached coral looks like. It's lost its zooxanthellae and is very evident as just this white skeleton. Most of those corals in August were still alive. If that bleaching continues, though, as Valeria mentioned, those corals will die. Just going to the next image then, similar side-by-side comparison from this past January to about a month ago on Sims Reef, another reef off of New Providence. This one, though, dominated by more of the mounding corals, Orbicella favulata and Orbicella annularis. But again, we see a fairly healthy coral colonies in the January imagery, contrasted to the stark white ones that are bleached in August this past year. We can also see in here a number that are still not quite white. If you go back to the previous slide, these are pale and on their way to becoming bleached. So if we went back out again this month, we'd probably see a lot more of those corals in white. And some of the ones that are white might be dying and start to show up as having a algal covering on them where they're being overgrown by seaweeds. So that's what we're looking at now. But I want to take a step back in time to see what we know about coral bleaching and the history of coral bleaching in the Bahamas and what impact it's had. So next slide, please. Starting in 1983, we have some information, and this is really the first regional bleaching event that was recorded during the 1983 El Nino. And we have some records from around the region, but for the Bahamas, an area called the Gingerbread Grounds, just north of Andros between Bimini and the Berry Islands, had extensive bleaching on brain corals there. But it wasn't something that was really followed up on, so we don't know too much more about that. However, when Lee Stocking Island, the Perry Institute's lab in the Exumas opened up the next year, we have a much greater record of bleaching events. So next one, please. The next bleaching event, so when it came in 1987, we have a lot more information, and Judy Lang did a lot of research there, describing that a greater range of species, more species were being affected by the bleaching, and that the bleaching was resulting in mortality, resulting in death for some of the species like the staghorn coral. Next one. There really wasn't much bleaching recorded then until 1998, and this was another big bleaching event that affected all of the Bahamas. We have very good data from the Exumas, where there was widespread bleaching, including bleaching of some of the soft corals, the gorgonians as well, and from Andros. And the work on Andros by Phil and Patricia Kramer and Brooke Gintert really was showing that we had mass mortality then. There was photo mosaic images similar to what I showed previously, going back to the 1970s for some of the monitoring stations off of Andros. And what Brooke found in her PhD dissertation was that there was about 25% or so, 20 to 25% of the corals were lost off Andros because of the bleaching event itself. They bleached and died through starvation, like Valeria described. But in the year following that, so throughout 1999, there continued to be a die-off of corals, and that's from disease. So the bleaching event made the corals more susceptible to diseases like black band disease that we generally only see in about 1% of the population, but about 25% of the population was infected and died there. So the net loss was about 50% of the corals for Andros at the monitoring stations. Next one. We have a few more bleaching events that we saw throughout the early 2000s. 2005 was a regional bleaching event, but it didn't affect the Bahamas that bad. Again, in 2010, next one, we saw a bleaching event in the Exumas, but largely the corals survived and recovered from that one. In 2015, then we saw another bleaching event that was more significant. In October 2015, surveys that we did off of Abaco showed 50 to 75% of corals across all species were bleached. In November 2015, we did surveys off of Andros Island, and 75 to 100% of corals were bleached at our survey sites there. Many of those did survive, but there was significant mortality associated with that 2015 event. Next one. 2019, we saw bleaching again in parts of the Bahamas, some of which was due to Hurricane Dorian. in the Little Bahama Bank and Abaco and Grand Bahama, but others due to heat stress down in the Exumas. And then next one. And that brings us to 2023, to the event that we're experiencing now. And we can see, going back in some of these past events, what is likely to happen based on the current bleaching event. But there's a couple important differences. One is that the bleaching event we're seeing now started much earlier in the year than previous events, July as opposed to August, September for most of them. The other big difference is that, as the slide Valeria showed with sea temperatures, it's much higher now than it was then too. And both the duration of high temperatures and how high those temperatures are will affect corals. Next one. And that's shown here in this slide. It's kind of confusing because there's a lot of colors here. The gray is land. The other colors are ocean areas coded by what's called degree heating weeks. So looking at how high above seasonal averages ocean temperatures are and for how long. So 1 degree Celsius above seasonal averages for 1 week is 1 degree heating week. 2 degrees above seasonal averages for 1 week is 2 degree heating weeks, or 2 weeks of 1 degree above average is also 2 degree heating weeks. So it takes into account both how high the temperatures are above seasonal averages and how long they persist. The take-home message here is that when areas reach about a 4 on this scale is generally when we see bleaching start to occur. When they hit around an 8 or so, it tends to be when we see more wide-scale bleaching and start to see some mortality. And on the next slide, if we zoom into the Bahamas, we can see in the Bahamas and Florida, we're kind of well beyond that point for most of that area. We're up in the teens to close to 20 in some areas for degree heating weeks. So severe bleaching with mass mortality. This has been widely reported from the Florida Keys throughout the summer in US news media. The Bahamas has largely gone unnoticed though, so we want to focus on that a little bit. But just want to stress too that there's a lot of areas in the Bahamas seeing this severe marine heat wave, but other areas like off of Grand Bahama and Abaco in the north, where we're still in the yellows, we'd expect much less bleaching. So what we've been doing, next one, is going to a number of these areas and doing different surveys, whether it's re-photographing our large-scale imaging photomosaic plots, or doing roving diver surveys, looking at both stony coral tissue loss disease and bleaching, or doing Atlantic and Gulf rapid reef assessment agri surveys throughout the islands. We can kind of see what is happening. And we've really focused in the central Bahamas around New Providence, Nassau, and Rose Island, around South Eleuthera, in the Exuma Keys, and in Abaco this summer. Next one. And what have we been seeing? Well, in June, up until about mid-July, we didn't see any bleaching at all. And water temperatures were getting a little bit high then, but we didn't see any bleaching. But all of a sudden in mid-July, it's like somebody flipped a switch. Around New Providence and Rose Island, sea temperatures were up to 32 degrees, 2 degrees or more above the seasonal average, and we started to see bleaching. About 10% of all species, mostly the species known to be very vulnerable to bleaching events, were the ones affected, like fire coral or staghorn coral. And anywhere from about a third to a little more than half of those colonies were observed to be affected. Next one. A few weeks later, end of July, beginning of August, resurveying those areas around New Providence and Rose Island, and also getting to Eleuthera and doing some surveys, Water temperatures were still very high and accumulating those degree heating weeks. And we saw more than half of the stony coral species and some of the soft coral species bleaching then. For some of the species that we had seen bleaching previously, up to 100% of colonies were already bleached. For other ones that were just starting to bleach, it was half the colonies or less. And we also started to see our first death due to the bleaching event then too. Next one. And this is just a video to show what some of those reefs, this is a reef off of Nassau, what it looked like at the end of July, early August during that time. So all of the staghorn coral on this reef was bleached at that time. Many of the other colonies you see are bleached or partly bleached or are starting to turn pale on their way to becoming bleached. So this is really kind of in the early stages of the bleaching event for this reef. If we went back today, we'd probably see most of those staghorn coral colonies dead, unfortunately, and a lot of the other colonies there starting to bleach more significantly. We can go to the next one. So that was late July into early August. Mid to late August, again, going back to the same places and going to the Exuma Keys, where we did some agri-surveys throughout the Exuma Keys Land and Sea Park. Water temperature still elevated, and nearly all of the coral species were affected. We did see on reefs up to, in the Exuma Keys, for example, all the reefs that we surveyed had 80 to 100% of corals bleached. And nearly all species were bleached. There are a couple species that seem to be a bit resistant that did a little better, but nearly all the corals we saw were bleached at that time and starting to see significant amounts of mortality. And then next one. And then that brings us to this month where, again, continued monitoring in the New Providence, Rose Island, and Eleuthera reefs. We're still seeing the same thing. We're still in this bleaching event. We haven't gotten through it yet. In Abaco in the north, we had a team go there in the end of August and then again last week. And it's still somewhat patchy bleaching, low mortality rates, lower number of colonies bleached, and some of the more resistant species having significant amounts that still are not bleached yet. So it's definitely variable across the Bahamas. And like that degree heating week map showed, or the coral warning map that Valeria showed, the northern Bahamas don't seem to be experiencing the same extreme temperatures in this marine heat wave that the central Bahamas are. Next one. So what does all this mean? Well, we're definitely seeing high mortality rates throughout much of the Bahamas across a wide range of species. We're seeing this happen now in real time. What's likely to happen moving forward as this bleaching event continues and in this aftermath? Well, based on what we saw in 1998, it's likely that the corals will be more vulnerable to diseases like black band disease. So we would expect an increase in diseases afterwards that'll lead to further coral mortality. And then this year, we also have stony coral tissue loss disease, and we don't really know what the interaction is going to be between the two, but we already are seeing additive impacts. So stony coral tissue loss disease has already had a devastating impact on many species on many reefs. Added on top of that, now we're seeing bleaching occurring and affecting a wider range of species even and over a greater area. So the situation is likely one where we're going to see major changes, like in the photographs there, where we go from live coral cover to greatly reduced coral cover on reefs. The species composition can change. And all that over the long term is going to affect the ecological function of coral reefs, and the services they provide. Now, when the coral dies, there's still that coral skeleton, that reef structure that remains behind. So some of these changes won't happen overnight. The habitat structure is still going to be there for fish and other things. But over time, as that reef erodes away, as new species come in and take the place of where living coral was in the past, we're going to see changes. So there's going to be some immediate impact on corals, and then probably lingering long-term effects over the next decade or more that are going to lead to significant changes on these reefs. Next one. So the situation is dire, to be honest with you, but that doesn't mean that we don't have some hope, and there's not some actions that we can take to help these reefs recover. And I'll just talk about this a little bit. There's a lot of reason for hope out there. And Valeria mentioned it, and I'll reiterate it with this photograph here, that bleaching isn't necessarily a death sentence. Just because a coral's bleached doesn't mean it dies. They can recover from it. Not all will. Many are going to die, but there will be some that survive, especially if there's a little bit of living coral, healthy coral tissue on there with algal symbionts. And this is from work that Dan Thornhill did at Lee Stocking Island in the Exumas after or during the 2005 bleaching event, where he tracked coral colonies over time. And many of them did recover from that, especially those that did have some places where the algal symbionts remained. So this just shows from September to November 2005 during the bleaching event and immediately after it. And then within a year, that coral had fully recovered. So that can happen. Next one. Oh, and this is just another example of that from up in Abaco, from Sandy Key Reef in the Pelican Keys Land and Sea Park. This is a huge Orbicella faviolata colony, about 3 meters across, hundreds of years old. I took this picture during the bleaching event in 2015, thinking that this colony was going to die, and this was the last photograph of it that I'd be able to take. But in May 2019, went back to the reef, and that colony had fully recovered, looked exactly the same as it had back in 2015 or before then. So some of these colonies will survive the bleaching and will recover. Next. The other thing is that these colonies are still reproducing. They're still putting out eggs that can be fertilized and can survive in new generations of coral. And this is a photograph that Natalia Hurtado, who works with us at the Bahamas Coral Innovation Hub at Cape Eleuthera Institute, took just this year during the bleaching event. Even though this coral was bleached, it still spawned. So if we make sure we reduce the effects of other stressors on corals, pollution and other things, if we help maintain ecosystem function that will give these next generation of corals a chance to survive, that we can have hope that these coral reefs will recover. Next one. There's also this case where we can see two coral colonies, same species, growing right next to each other, subject to the same environmental conditions, the same water temperature, the same light levels, the same other stressors, whether it's pollution or other factors. And we see very different responses. One of these is completely bleached. And to be honest with you, is if things continue the way they are, is likely to die because of the current marine heat wave. But the one right to its left, a little pale, yeah, but it's doing fine and has a good chance of surviving. So there are some differences among these corals that make some more resistant or more resilient to these sorts of events. And as scientists, we can then look into that and see why and try to harness that information. And that's just what we're doing. Next one. This was a paper we published a few years ago about some reefs off of Abaco. The picture on the right is the same one I showed you before from Sandy Key Reef in the Pelican Keys Land and Sea Park. And the photograph on the left is Mermaid Reef, which is right off of Marsh Harbor. Both of these reefs went through the 2015 bleaching event. Sandy Key Reef had extensive bleaching on it, though, especially for this particular species of coral, Orbicella favulata. But Mermaid Reef, the one on the left, didn't bleach at all. Next one. And that was really amazing to us because Mermaid Reef sees temperatures that are so much higher than Sandy Key Reef. That graph on the top left, the pink color, or both ones, the pink color is Mermaid Reef, the bluish green is Sandy Key Reef. And for Mermaid Reef, from about middle of June until end of September, beginning of October, the temperatures were above that kind of bleaching threshold, that 30 degrees for almost the entire time. Sandy Key Reef kind of went above it sometimes, dipped below it, did not have nearly the degree heating weeks that Mermaid Reef had. And yet Mermaid Reef didn't bleach at all, Sandy Key Reef did. So what's going on there? Well, for Mermaid Reef, we did a lot of or for both reefs, we did a lot of studies looking at the genetics of the corals, looking at the zooxanthellae, looking at the microbiome, the other microbes associated with the corals. And we found some significant differences between those reefs. On Mermaid Reef, all the corals in that picture there were 100% identical genetically. So they're basically all clones of each other that perhaps are uniquely adapted to living in those extreme temperatures, versus Sandy Key, where we had a greater diversity of corals. Next one. We also saw, and I know this is a little bit difficult to interpret, but looking at the zooxanthellae on Mermaid Reef, the column on the left here to the left of the dotted line, were all the genus Duristinium, a genus of zooxanthellae, so the microscopic algae living in the coral that's giving the coral its energy. This particular genus is known to be heat tolerant compared to the other ones. So again, we have Mermaid Reef that only had this heat tolerant zooxanthellae. Sandy Key Reef, it had mostly other less tolerant zooxanthellae and bleached. There was also differences in the other microbes on the reef too. So we can identify what these differences are. Next slide. Identify what these differences are, whether it's differences in the microbiome, the genetics, or other chemistry of those, or biology of those corals, that make some more resistant or more resilient than others, whether it's resilient and resistant to disease like stony coral tissue loss disease or to bleaching, and see if we can identify solutions based on those differences, whether it's applying a microbiome, microbial treatment that will make them more resistant to disease, or whether it's selectively restoring reefs with ones that are more heat tolerant. We can help jumpstart the recovery process and help build resilience in these reefs through science. Now, we're only going to be able to apply this over a certain amount of area. It's still going to be a little bit experimental, but it lets us identify these areas where we can make an impact. And by making these impacts incrementally, we can have a greater impact on the recovery of reefs after these bleaching events. Next one. So we do have a path forward. I think there's some very important steps that we can take. We can't stop the current bleaching event, but we can reduce other stressors that might make corals more vulnerable to the heat stress, or will prohibit their recovery if we allow them to happen. So we can control things like pollution. We can do some things to control predation or disease on these reefs. We can maintain natural ecosystems in parks, protected areas that may build in the natural mechanisms that help reefs recover, increased grazing, whether it's from fish like parrotfish or invertebrates like urchins. So there are some things we can do on that front. That might only get us so far, though. So I think it's worth also investing in the research to identify resilient corals, and then to help kickstart the recovery process by using those resilient corals for restoration or rehabilitation on the reef. But really, the big thing that we're faced with, it can reduce the extremity of these marine heat waves and reduce the frequency, is related to climate change. And that's going to require government action on a global scale to reduce the impact of climate change. And until we do that, we're going to be stuck with these patchworks of other solutions that we can try to apply. That's really the thing that's going to make the biggest difference in stopping the effect of the marine heat waves. Next one. And I guess we'll end it there. We'll be here to take questions for both Dr. Pizarro and myself. And thank you very much for listening.
Thank you, Dr. Dahlgren, and thank you, Dr. Pizarro. So before we get to the question and answer portion, okay, the link is in the chat. Please complete this quick audience survey and feedback form. It's fairly short, but now we will move on to the question portion. So you can type your questions into the Q&A option, or you can also raise your hand to ask verbally. But the first question is, how can a non-biologist diver tell the difference between bleaching and stony coral tissue loss disease?
I'm gonna take that one, Greg. So it's a matter of kind of educating your eye, right? So you have to, there are two things. Again, diseases you will see coral mortality. So when the coral dies, the exoskeleton is exposed. So you have to get really close and you have to see like all the different characteristics of the exoskeleton to be able to tell that it doesn't have any living tissue on top of it. With a stony coral tissue loss disease, you can have some area where the disease, like where the border of the disease is, that is pale or bleached. So you will have like dead skeleton, exposed skeleton, this bleach part, and then living tissue with the zooxanthellae. Bleaching, you will always have living tissue on top of the exoskeleton, but it will be like the colony, most of it will be white. So it's mostly just learning how to see the living tissue that is very thin in many species.
Okay, thank you, Dr. Pizarro. Our next question is, looking at the coral individuals in your last slide growing on the ropes, I was interested in reef restoration practices that you have at the moment, and how are they going?
All right, I'll take that one. Right now, we are working throughout the Bahamas and some other islands in the Caribbean on coral restoration, using corals that were take cuttings basically from healthy corals in the wild, and are growing them in open water nurseries like the one that I showed, different forms of nurseries. It's going quite well. We have around 10,000 or so corals being grown in nurseries at present. We have done this for over a decade in the Bahamas and have definitely improved reef health in terms of restoring some critically endangered species, increasing living coral cover, improving habitat for fish and other things. But those corals, just like the rest of the reefs, are being hit with this marine heat wave right now. So we are seeing loss of some of those corals in nurseries and ones that have been planted out on the reef due to the marine heat wave. What's interesting, however, though, is that we have corals that are from areas that see chronic heat stress, that see very high summer temperatures year after year after year. Those corals in nurseries are doing a lot better than ones that are from areas that really don't see that heat stress. So again, there's science that we can do to help identify which ones are going to be more resilient, and we can, moving forward, target those to improve the success of the nurseries in the event of an extreme event like we're seeing right now.
Alrighty, the next question is, I'm in the Keys and go out weekly to clean the algae around the base of our outplanted coral, and we use toothbrushes. Is this helpful, or does this add extra stress?
I'll
So
take that one too,
you take that? Okay.
unless you want to. So I think the answer is it depends. As long as you are not brushing the coral itself, it should have a positive impact. We do know that there's pathogens that are in the turf algae and stuff on reefs, in the seaweeds, that can affect corals, or trap sediments that can affect corals. So by doing that, you can help the corals a little bit. You do run the risk, if you do brush over the corals, of damaging them that way. And in a lot of cases, the corals can do quite well even without that. So I don't know if there's been definitive studies on it, but it may help, but it might not be necessary. And Valeria, do you want to add to that?
I would say that it depends on what kind of algae you are removing. So I don't think that it has any extra stress if you don't brush the coral tissue. But in some cases, removing especially like cyanobacteria can be good on, or hydroids, because they will fight the coral too. So they will prevent them to grow.
Okay, thank you. Next question. What do you consider to be the most urgent research priorities in the coming year?
Go ahead, Valeria.
Oh, well, research-wise, of course, and this is something that researchers in other places in the Caribbean are doing, like kind of trying to identify like what are the pathogens for the different diseases, so we can find ways to cure or stop the diseases in a more effective way, and especially more scale, because we do have something for Skittle, but for many diseases we don't have anything. Identifying, as Craig said, like resistant corals to bleaching, to diseases, being able to work with different methods of coral restoration, coral propagation, larval propagation, all this. So there are so many things that people is doing in other places. We think that it will be very important, especially for the Bahamas, to be able to do many of the genetics that right now we cannot do. But a lot of, I will say that we have so many things that we need to do, but maybe just to understand like all the genetics of the corals and the microbiome, so we can scale coral restoration and have the corals not as bad as they used to be in the 80s, but functional for humans and to keep existing in the Bahamas.
Yeah, and just to add to that, we do have a lot of advanced tools and technologies in the field of genetics and other things to really help with this. There's a lot of international expertise that is needed to do that, because it's a very specialized field, and there's only a handful of people in the world doing this work. So anything we can do to encourage those kind of international collaborations that involve both local scientists and specialized experts is really going to be helpful in advancing our science and finding cures or solutions to diseases, to bleaching, and to improve restoration.
Alrighty, next question is from Rosalie. Based on previous bleaching events in the Bahamas, can an estimate be predicted for how many colonies may revert back to a healthy state from this year's bleaching event?
I would say, unfortunately, it's a little too early to tell. We're still right in the middle of this bleaching event. And like we said in the talks, we're a little bit in uncharted waters here. We haven't had anything this bad to really refer back to. So the closest we have is 1998, and we know that in some places killed up to 50% of corals. We could be faced with something similar to that right now. On the other hand, some of the ones that survived 1998 that have been breeding since then might be some of those more resistant ones, and it might not be quite that bad. So we just don't know. But we do know the 1-2 punch of both the bleaching and stony coral tissue loss disease puts us in at risk for severe loss that may take decades or even centuries to recover from.
Okay, next question is from Stacy. She says, very interesting data. To build on the differences between Sandy Key and Mermaid Reef, you mentioned that Mermaid Reef had all one genus of algae and Sandy Key had 2. We know that Sandy Key coral recovered from the bleaching. Do you know if the recovered coral has a different composition of zooxanthellae than it did prior to bleaching?
So a couple things there. Not all the corals from Sandy Key did survive. So that's one thing. The other thing is that the sampling that we did of tissues from those corals to look at the genetics of the corals, the microbiome, the zooxanthellae, was all done after that bleaching event. So we don't know how diverse it was before that bleaching event, how much was lost during that bleaching event. We only know what made it through that bottleneck, unfortunately. So that kind of goes to the point of we need to be developing this database of genetics so that we can see how things change over time. We just didn't have it at that time. We didn't have the tools to do it at that time before that bleaching event.
Rick asks, what can be done to increase fish populations and diversity that help to reduce algae cover that prevents coral settlement?
So I'll take on that one, Valeria, again. So the fish that are most responsible for removing algae and potentially increasing the ability of corals to settle and survive are parrotfish. So I think the biggest thing is not fishing for parrotfish. The Bahamas has healthier parrotfish populations than most parts of the Caribbean. Parrotfish haven't been as targeted in the Bahamas as they have in other parts of the Caribbean. So the Bahamas already has a head start there, but we are seeing increasing consumption of parrotfish, increased fishing for parrotfish, and that's of concern. So protecting that species of fish in general is an important thing. The other thing is it's not just the fish, it's also invertebrates. There's some crabs that eat a lot of seaweed, also sea urchins, and those are also under threat, whether it's from disease or from, in the case of the crabs, potentially fishing for them. So we do have a need to protect those and help those populations build back up too. And Valeria, I don't know if you want to add anything to that.
No, I agree. Like, stop fishing, especially parrotfish and crabs.
Okay, so for our next question, there seems to be a trend for a few of these questions. I think the answer may be somewhat applicable to all of these questions. So I'll go ahead and just fire off a few questions because they're all related. So Douglas wants to know, is there a plan to expand the reef rescue diver specialty, like provide certification to PADI aware instructors? Samantha is asking, is there someone I can speak to that can provide guidance on what type of engineering and what career path would be best to enable me to help protect the marine environment? And Michael asks, to you, what role do trained volunteers, corals, and monitoring certified divers play in coral restoration? How can a group of volunteer divers plug in and help the Samantha says, how can we get involved in the process of restoring corals and marine ecosystems in the Bahamas without having much experience in the field?
All right, so I'll start with that, and Valeria, please feel free to jump in. But I think things that anybody can do. It's great that people want to get involved, and there are a number of things that can be done. Like we were just getting at with the last question, choices that we make in terms of what seafood we buy, what we consume, make a difference. So anybody that eats seafood has a role to play there. So avoiding things like parrotfish or the big channel crabs or king crabs that people call them are important steps that can be taken that help the natural resilience of reefs to recover from impacts, whether it's disease or bleaching, is something that anybody can do. There are also opportunities for people that are scuba divers to get involved with restoring coral reefs. And we've created the Reef Rescue Network to do just that. And at present, we're working with, I think, over 80% of the dive operators in the Bahamas, as well as dive operators in several other countries around the Caribbean, to train recreational divers to restore coral reefs, to grow corals in nurseries, plant corals out on reefs. And we will shortly be launching a new website, and somebody can put it in the chat to give you the address, that will help put you in touch with those dive operators as part of the Reef Rescue Network. So there are opportunities there. There's going to be increased opportunities moving forward as we expand and build on that network so that we can get more people out there helping to restore reefs. Valeria, do you want to add to that?
Yeah, I would say for Samantha that it was asking about like kind of how to approach like the marine conservation problem throughout engineering. It's like, Samantha, if you can go to our webpage, it's just there in the presentation, and just like look our profiles, or our emails are there. You can contact us and we can just schedule a call and talk about like the different perspectives from our marine science point of view. You're muted, Danielle.
Thank you. Okay, next question is from Crystal. She says, excellent presentation and knowledge sharing. How long will it generally take corals to bounce back on their own after the water cools and the heat goes away? Will bleaching occur every summer and then bounce back during the cooler months naturally?
So depends on the species, depends on how long the temperatures have been high. So some species can bounce back very quickly. And for some, it will take a little bit longer. The key will be in how fast the temperature goes down, like in the case of this marine heat wave, and that stays in that lower preferable temperature. That is, for corals, they really like the water temperature to be 28 Celsius or 82 Fahrenheit. So like with the, I don't remember the name of the hurricane that passed through Florida. So Florida had been having this massive bleaching event. They had this hurricane passing through. The temperatures went down, but it was only for a couple of days. So it wasn't long enough for the corals to recover. So it's more a matter of that. If you have like a really, sometimes, especially last year, we had a really minor bleaching in some reefs around New Providence, and the corals recover in less than a month. I will say that from this bleaching, it will probably take a little longer. But again, it depends on the species and depends on all the factors, like how long they were bleached and how long it takes the temperature to go down.
Yeah, I would just add to that. Over the past 5 years or so, we are seeing some bleaching on an almost annual basis. So, and the corals recover fine from that. It's really just these severe events that we need to be particularly concerned with. And the ones that recover from this one might suffer what's we call partial mortality, where part of the coral colony dies, just like a tree. Several branches could die, the rest of it's still alive and can survive. We are likely to see that with many of the survivors in this. So maybe part of the coral dies, the rest survives, and then hopefully we'll be given the opportunity to continue growing and overgrow some of the dead area over time.
Thank you. Before we move to our next question, I just want to remind the participants to fill out the feedback survey form that was placed in the chat earlier. So our next question is, are we seeing any other species in the Bahamas that host zooxanthellae as symbionts? And if so, are they bleaching as well? Or is there anything we can learn from these animals if they are not bleaching?
So corals, most of the hard corals that we are seeing in the reefs in Bahamas, they are bleaching. Some other animals that they do have zooxanthellae, like some sponges, some I forgot the names, some soft corals, not hard corals, but soft corals, they are getting bleached. We have seen, especially for some sea fans, that they are dying, and we think that is temperature related. So they are, and again, like they will bounce back if the water temperature goes down. Craig, do you want to add anything?
No, I'll just say that one of the first organisms to bleach that we saw this year was a soft coral called Palithoa. And that was really, that and the fire coral were some of the indicators that this was going to be a severe bleaching event. We know from the past, from 1998, there was a researcher, Howie Lasker, who studies soft corals, that did a lot of work in the exumas, and he documented which species of those that have zooxanthellae bleached, and how severe that bleaching was, causing death for some of them. So we do have a little bit to go off of there. But a lot of those that do have the zooxanthellae suffer the same impacts that the stony corals do.
Okay, next question is, has there been any studies to this extent in other Caribbean islands, or would there be any future studies in other parts of the Caribbean? And a related question that was asked as well is, are there any future plans to expand to Jamaica?
So I'll take the first part, and then Craig, you can talk about Jamaica. So yeah, like this marine heat wave has been affecting many areas like Florida, reefs, Mexico, Jamaica, Dominican Republic, Puerto Rico, many places like the Southern Caribbean. It's a little less affected by the moment, but they are expecting to get it. And they're doing different things, not only what we're doing, that is just like assessing and reporting and having numbers in terms of what species have been affected, how many corals are dying. Like in Florida, the mortality is very extensive. At the moment in Mexico, like the elkhorn and the staghorn, they have lost already 70% of the populations, the white populations. And there are some researchers that are doing studies in terms of trying to see which ones are the resistant ones and see what is the zooxanthellae that they have, the symbionts, the genetics of the corals. So this is something that every researcher all around the Caribbean is keeping an eye on, because they expect this to be a major bleaching event for the whole, the whole Great Caribbean.
Yeah, and just to expand on that, we were part of a consortium of researchers from different U.S. universities and research institutions working in the Florida Keys, looking at how different colonies of staghorn coral are more or less tolerant to different temperature ranges in the dry tortugas area of the Florida Keys, doing a lot of experimental and outplanting to different areas with that. And unfortunately, we had a big research cruise as part of that project in June, setting up experiments. By the end of August, all of those corals had died from heat stress. We documented differences in survival, but the extreme heat stress overcame all of those. So it can be difficult to do this kind of research at times. The other thing I want to add as far as expansion to other parts of the region, we have had some projects working with partners in Jamaica recently. I think Valeria was just down there earlier this year. There's an awful lot of work to be done throughout the Caribbean, and the Caribbean provides such a diverse environment to look at how different disease spread or temperature conditions can affect corals and reef development. So the more we can do on that kind of regional basis, the better. And that's, I think, something that we would really like to get more involved with, working with local partners in that country.
Okay, Anna asks, this is kind of a two-parter. We're seeing a huge amount of Skittle D spreading on Andres. Have you had any success in treating Skittle D, and is there currently any training programs available? Next part is also, if interested, I would be happy to speak to someone about providing bleaching/disease information for Andres, as I noticed that it is one of the areas you listed as needing more info.
Okay, so yes, we know that Andres, we have done assessments in Andres to know the extent of the spread of Skittle D, and we have treated corals in different parts of Andres. We try to go as often to different areas through these that sometimes our team is not big enough to cover all the reefs in the Bahamas. So we do have some prioritized areas, and we know that treatment is effective. We have to treat corals as much as possible. But Anna, I would say, like, again, go to the webpage, send me an email, and we can talk about like how to get you more information about coral bleaching, coral diseases, and see the possibilities about training. We are doing some trainings. I have to say that I would like to do more. It's a very time consuming, and sometimes we don't cover as much as we want.
Yeah, and I'll just add to that. We definitely are interested in working with people locally on different islands as much as possible. And we do have some potential training opportunities moving forward. But even just photos or anything is extremely helpful because, like Valeria said, we can't be everywhere all the time with the team that we have. So that would be great. And then as far as the treatments go, right now, the treatment for stony coral tissue loss disease involves treating individual colonies with an antibiotic paste or putty, basically. And it's extremely, it's effective. It's over 80% effective, but it's not cheap. It's not the easiest thing to do, and it takes a lot of time. So we have some limitations on how much we can do, and we really want to prioritize areas. So we can't do it everywhere, but there are places where it might be a good line of defense to help curb the spread of the disease or protect certain coral colonies or certain areas. So definitely send us more information and we can have a conversation about that.
Okay, Jules asks, lessons learned in your experience could be a good stepping stone for other institutions like here in Coron, Palawan, Philippines. Could you provide more feasible methods of predetermining bleaching events using viable biological indicators?
You want to take that? I can take it.
Well, I'll just start by saying I think the best predictors of bleaching are sea temperature and climatological models. And NOAA, National Oceanic and Atmospheric Administration in the US, does have products available online for most of the coral reef areas, I believe, that can be used to at least be a rough predictor of coral bleaching. You go ahead, Valeria.
Yeah, no, they have the tools. They have one that is like focused on the Atlantic, but they have another one that is like kind of worldwide. So they have like watch alerts, and you can subscribe to their emails and just like kind of look at your region. And from that information, when they just send you a watch alert, that will mean that is that yellow color that I show you in the last map that I show. And from there, you should just start going out as often as you can to see if you get any pale. The first thing usually is like you get pale corals. So they will like kind of look a little more pale than the usual color. And maybe you can monitor some specific colonies and see how it is and move from there. And if you get the possibility to identify corals that are not bleaching, I will say if you can just tag them or follow them, and that will be the start.
Okay, our next question is from Erin. Have you experimented with shading or feeding bleached corals? If so, does it seem to be helping, and can it be applied on a scale that has true impact?
Yeah, the main problem is really scale. Like you can do that, especially for coral nurseries. You can put some shading, or you can move, in some cases, corals from shallow waters to deep waters. The problem that we have with the present marine heat wave, is that the water temperature is still very high, even at deep, at the depth, in more deep areas. So you can reduce the UV stressor with the shade, but like with this marine heat wave, it won't do much. So you can feed corals, but to feed corals in a reef scale, it's very difficult. So you can do that if you had them in confined environments, but in an open water, feeding them, that will be really difficult. And we went to a night dive, and I was trying to feed as many corals as possible, just putting the dive light on top of the coral. I think that I saved like 10 corals for a week.
Okay, we have had a few questions that asked basically, what is the Bahamian government doing to combat climate change?
I'll take that one. So the Prime Minister has definitely been at the forefront, representing Caribbean states and small island developing states, in speaking out to raise global attention among the governments of the world about climate change and how it's affecting the Bahamas. So there's definitely a call to action out there. Unfortunately, and the government of the Bahamas is also taking some steps locally that may be allowing more climate change prevention measures to go in place related to sustainable energy, things like that. Unfortunately, the Bahamas, being the small population that it has, those steps are only going to go so far to solve a global problem. It's really some of the bigger, more developed countries that need to act. And the Bahamas, unfortunately, is subject to what those countries do. The Bahamas also recently has passed legislation related to blue carbon, related to the sale of carbon credits and offsets. And that may be a mechanism for the Bahamas to play a role on a global stage. The Bahamas does have resources, mangroves, potentially seagrass beds that are big in terms of the amount of carbon they can sequester out of the atmosphere to reduce the impacts of climate change. Whether or not those measures are even effective is somewhat debatable. As far as the sale of carbon credits and offsets, how the Bahamas government is implementing that is still yet to be seen. So there are some potential ways the Bahamas can be a player in terms of fighting global climate change, but it's difficult for such a small country to have a big impact.
Okay, next question is, why is there less heat stress in the northern Bahamas, but more heat stress in the Florida environment?
So, so there are a couple of factors, right? And especially with these marine heat waves. First, well, it's a little more north and more exposed to the Atlantic. So it has more influence from Atlantic currents, if you can say. We have to say that there have been different storms and hurricanes like Lee, that even though it passed a little far from the Bahamas, it did influence a little bit those, like cooling those waters. So that is actually what is happening in that part of the Bahamas, which is great because they have been affected by stony coral tissue disease. So it's good to have not that much stress in that area.
Yeah, I'll add to that too, that a lot of these conditions are, to build on that, very local. The shallow Bahama banks heat up a lot. If anyone goes diving or swimming even, heat up a lot in the summer and cool down a lot in the winter. They take on the temperature of the atmosphere faster than deeper water areas. So the Bahamas has so much shallow bank area that that it does heat up very quickly and has a big impact. But there's also factors related to wind and the atmosphere. If the wind is blowing a lot out of the south, like it has been for a period of time, it can cause upwelling, deeper waters from hundreds of feet down to come up to shallower areas and cool things a little bit, or hurricanes like Valeria mentioned. So it's a little bit complicated in how the atmosphere and ocean interact, and how that influences temperature. And I think in the northern Bahamas right now, it's a combination of several things that are keeping temperatures a little bit cooler than some of the other areas.
Okay, next question is, given the observed loss of species diversity in coral ecosystems with a greater survival of heat-tolerant corals, what are some of the impacts you expect to see down the food chain within the Caribbean?
I'll start and then you can talk, right? So in terms of corals species diversity loss due to bleaching or something, the truth is that we haven't lost like that much diversity. We have some species that are highly susceptible to die, and we might have like local extinctions, but we haven't lost, like in terms of corals. We have the main problem is like the changes in the community composition, and that can actually affect the functionality of coral reefs, and that is the main problem. So if we have like very low coral cover, what we're going to have is more erosion. And this, as Craig mentioned at some point, it's going to take years for the coral erosion to like kind of flat the coral reefs if we don't have more reefs, and that will have an effect. Like it's like a snowball effect. It will start just grow and grow and grow and affect, like at the end, us, right? But it's not that losing the diversity is not bad, but it's a little bit more complicated, and it involves just like all the cascade of effects that it will have to reef functionality.
Right. And just to build on that a little bit, because there's not many species of fish or even of invertebrates that actually feed on corals, you don't see that change immediately. It's more a change of how the habitat structure changes as the reefs start to erode or deteriorate. And with less complexity, we see it's difficult to predict which species in which areas are going to be affected more than others as far as fish and things like that. But we do know from a number of studies is that the overall diversity is going to go down, and will likely have a significant impact across a broad range of species as well.
Okay, the next question is from Rick. Have you seen cases where areas of sparse staghorn coral growth over many years have all of a sudden exploded in growth? I am located in Salt Key. This is south of Grand Turk in Turks and Caicos. And I found 2 sites on the wall edge, approximately 35 to 40 foot deep, and have expanded very dramatically. Would this be something that would be interesting to study?
Let me take that to start with, Valeria, and you could add it. And the short answer, the second part of that question is, yeah, it would be very interesting to study. Staghorn coral and its close relative elkhorn coral are 2 of the faster growing species of corals. They're also ones that their populations have been knocked down considerably. So we've been very much involved with restoration on those species, and it has been amazing to see when we can get the populations reestablished, how quickly they can expand their area, how quickly they can grow. The downside of that too is that they can quickly get wiped out by bleaching like we're seeing now, or disease like white band disease that can come through occasionally. So they definitely are ones that it's great to see it when they take off, but I always am a little cautious about what's around the corner that might affect them later on.
Yeah, I will add to that, like they are, as Craig said, the fastest growers. And the other important thing, and that's why when you have like good populations, they can really reproduce and expand really fast, is that while doing fragmentation, they're the ones that can just like kind of reproduce. So when you have that, is you have very low genetic diversity. So when you have diseases, they can die massively very quick as well.
Alrighty, Timothy asks, has anyone had success with planting frags in deeper water?
The short answer is yes. The more complex answer is you can do it. There are some species that will respond better than others. It's usually for corals, it's usually better that when you are taking them from shallow waters, don't go straight to very deep waters, because the changes in the environments can be very stressful and you will have like higher mortality. So what some people does is they take them from shallow waters and they do like slowly take them down. But yeah, it is possible and people has done it.
Yeah.
Okay, our next question is from George. This one is slightly long, but the work presented seems to be focused on identification and selective breeding of tolerant corals to repopulate the reef with the same. Would that serve to limit genetic diversity on resulting reef populations, albeit with more resilient corals? And what are your thoughts about more random sexual propagation and reseeding, resulting in greater genetic diversity and letting natural selection determine the survivors? I realize we can do both, just wondering about your thoughts.
Yeah, no, and I definitely agree with the latter part of that is, I think it is important to do both. If we limit genetic diversity, we don't know what we're losing in doing that. The one thing I will say though is, especially in this current bleaching event, if we don't do the selecting ahead of time, nature's doing it for us afterwards, and we're wasting a lot of time and effort on corals that are not going to survive if we don't try to select some of the ones that are more tolerant. And looking at future scenarios of sea temperatures, we're only going to see more of what we're currently seeing right now. So we do want to try to preserve diversity, and there's not necessarily a one-size-fits-all approach because different reefs see different environmental conditions. But the ones that we see from the thousands that we're growing, that are the ones that are surviving, are the ones that are from colonies that are in areas of chronic heat stress that have adapted to living in those conditions moving forward.
I'm going to add to that I, and in the way kind of, I agree and disagree with not just like kind of selecting the ones that are doing better. I do think that the things that we think that are the weakest, they could be the best in terms of diseases. So I will say, like, yeah, we try to do everything. I do think that for thermal stress, we focus on the ones that are living more, but as you say, like working with larvae and coral sexual reproduction, that will give us everything. And we need to kind of have a broad, as much as we can. That is the thing, like as much as diverse genetic diversity we can have is the better.
No, I definitely agree with that. The other thing to think about too is that it's not just the genetic diversity of the corals, it's also looking at the holobiont. So making sure the microbiome, the zooxanthellae are also incorporated into that. And the ones that might be resistant to stony coral tissue loss disease might be the ones that are vulnerable to heat stress and vice versa. So we need to look at it and not just screen for one factor, but screen for multiple factors if we are going to go down the road of selective breeding.
Alrighty, our next question is from Zoe, and she wants your professional opinion. We are also seeing a lot of non-coral growth on our domes. They're made with metal. Her question is, would PVC domes be better as they would be easier to clean? The algae is quite difficult to remove from the metal.
I'm gonna say from my experience, I know that at the beginning, especially with the metal domes, you have a lot of algae, but in the long term, if you have coral surviving, they will do much better in the long term from my experience. I had some PVC domes in the American Pacific, and they did well a couple of years, but the PVC being plastic starts to degrade and they break more easily up after a couple of years. So I will stick to the metal ones.
Alrighty, it seems as though our questions are winding down, so I will just ask the final few. This one is, what is the condition of the reefs on the Andreas Barrier Reef?
Right, do you want to take that one?
Sure. So I'll start off with a caveat that it's been a little while since I've done an extensive study of the whole Andreas Barrier Reef. I will say that it varies from place to place a lot in terms of historically what it once was and currently what it is now. I would say in general, the reefs there have been doing pretty well, although we are seeing the impact of past bleaching events, and in places, stony coral tissue loss disease, which is expanding throughout the Andros Barrier Reef right now. So yeah, I would say the condition there has been good. The double threat that they're faced right now could change that very rapidly, though. And let me also just add before Valeria, you chime in. By good, that's a relative term, right? They're definitely not what they once were. They're definitely not what they were 50 years ago or longer than that. But compared to other reefs throughout the Bahamas, they've fared better recently.
Yeah, my experience in Andros, it's very limited. I'm gonna say I've been in South Andros and North Andros and not in between. And Andros Barrier Reef is so big. But I agree with Craig, like the condition, especially in South Andros, when we haven't had stony coral tissue loss disease, is much better than now in the north. They're just like kind of getting the disease and it's just spreading very fast.
Alrighty, and our last question is from Michael. Is removing fireworms from corals worth the effort to reduce the stress as well as the snails?
So I'll jump in to start with that, Valeria. Yeah, if we're talking about things like staghorn coral that are being used in restoration, two of the biggest factor that we've seen in some parts of the Bahamas and other parts of the Caribbean with jump starting their recovery is predation, whether it's by fireworms or the snails, Corallophilia, Galea. And removal does seem to be effective. It's very labor intensive. You can only do it on relatively small spatial scales. But if it's to help the recovery process with particularly staghorn and elkhorn coral, I think it is worth doing that investment until those populations are large enough where they can outgrow the threat of that predation. Valeria?
Yeah, I will say yes. And fireworms, especially in places when you have like an outbreak, like in some places when you have like one or two, I wouldn't do it. I will do it in places where you have like, yeah, an outbreak of fireworms and that happens. Or if I see a huge one just eating all the staghorn. And with snails, yes, and especially because now they've been linked a little bit of the spreading the stony coral tissue loss disease locally. But as Craig said, it's a lot of work and sometimes you cannot identify them. You have to have an eye and experience with, especially with the snails.
Yeah, and on that note too, with the snails, the main predator, well, there's a species of snails, Corallophilia, used to be abbreviata, now Galea, whatever, that eats corals. And that can have a major impact, especially on staghorn and elkhorn corals. And they'll move off of other corals to go to those. Removing them from corals, I would encourage. However, there's another species of snail that's the main predator on those that can control their populations that looks pretty similar unless you know what you're looking for. So if you just go and remove all snails, you may be doing more harm than good in the long term. If you can distinguish between those two and email me and I can help you send some photos to help with that. If you can distinguish between the two and leave the snails that control the predatory snail populations that affects corals, then it could be something positive.
Maybe adding to that, of course, that just don't do it when you're not allowed to take anything out of the water. That's like permit wise, everything, like, yeah.
Yes.
Alrighty, thank you so much, Dr. Dahlgren and Dr. Pizarro. And in closing, thank you all so much for joining us today to learn more about Coral Reefs in Crisis, a mass bleaching event in the Caribbean. For more information, please visit our website at theperryinstitute.org. And also, please follow us on our social media channels at Perry Institute for Marine Science, including Facebook, Instagram, Twitter, and LinkedIn. We hope you join us again in the future for our future webinars. Thank you again, and please enjoy the rest of your day.
Thank you.
Thank you. Bye.